Now we are in this section. We will learn about the methods of rDNA transfer. Okay, so the DNA, how we can transfer the recombinant DNA into a competent host. Okay, we have learned what is meant by a competent host. So now we are going to talk about what are the methods by which we can insert the recombinant DNA into a competent host. Okay, so there are different methods are there, but we have to study only the direct transfer. Okay, so direct transfer means what? It's a kind of method for transferring the rDNA into the organism. But what we are doing is that here it is directly inserted into the organism, into the host. Okay, so there are different methods. We have to study only 3 methods of direct gene transfer. Okay, and all these methods are physical methods. We are not talking about any chemical methods. Only 3 physical methods. The first method is the heat shock. First one is heat shock, another one is micro injection, and third one is gene gun or bioballistic. Okay, so this heat shock, it is otherwise known as temperature shock. So what we are doing is that this. So you just look at here. The host cells, they are incubated with recombinant DNA. Yes, first we are taking the beaker or any kind of petri dish. So we are incubating the host cells, that is the bacterium, with what? Recombinant DNA. What is a recombinant DNAs? Okay. So in a solution containing calcium ions. So this is the solution containing calcium ions, and we are putting some ice cubes there. Okay, around ice, and it will be followed by, first we are taking the petri dish, host cells are there, we are allowing them incubate with the rDNA. In the medium, what? Calcium ions are there, and there is ice. Then immediately after that, after some time, we are just removing this into what? 42 degrees Celsius, at least for 2 minutes. Okay, and this will give what? A sudden heat shock. Okay, it will give a heat shock to all these bacterial cells, and because of this heat shock, what happens is that some of the pores, they may be created. Pores in the cell membrane. Pores may be created in the host cell. Okay, when these pores are created in the host cell, what will happen is that this recombinant DNA can pass through this cell membrane and it will reach in the host cell. Okay, so that is one method. So what we are talking is the transferring of this rDNA into the host cell. Okay, so we are talking about only the direct method. That means we are directly injecting. Okay, we are directly inserting the DNA into the host. So here first method is heat shock. Another method, that is what? The micro injection. Okay, so in micro injection, the rDNA is directly injected into the nucleus of the host cell. Okay, so this is the nucleus of the host cell. Okay, so this is nucleus of the host cell. So in micro injection, what we are doing is that we are directly injecting the recombinant DNA into the host cell by using a pipette, that is fine micro pipette. By using a fine micro pipette, we are inserting directly the gene or the recombinant DNA into the nucleus. Okay, and it is used to transfer the gene directly into the oocyte or eggs or embryo. In the embryo or in the eggs or in the oocyte, we can directly inject the recombinant DNA. Okay, so that is the second method. Another one is what? The gene gun, or it is known as the bioballistics. Okay, here what we are doing is that we are taking some microscopic gold or tungsten particles. Okay, microscopic. Just look at this. Gene gun or bioballistic. So these are the microscopic golden particles or tungsten, and it will be coated with recombinant DNA. Okay, now this structure, it may contain what? gold particles with recombinant DNA. Okay, and we are firing this at the cells using a compressed air gun. By using a compressed air gun, we are firing these gold particles. What is the usage of these gold particles? They can penetrate easily into the cell wall. Okay, so with these gold particles, microscopic gold particles, the DNA is coated with that. Okay, so with these gold particles, the DNA will also reach into the cell. Okay, so these are the three three type of direct transfer of what this recombinant DNA into the host cell. First one is what? The heat shock. So first we are taking that host cell with recombinant DNA, calcium ions are there, and in the ice. Directly we are suddenly we are giving a heat shock that will create pores and recombinant DNA enter. Second one, that is micro injection by using a fine micro pipette. We are inserting this DNA, rDNA, into the host cell. Okay. Then comes what? The gene gun or biocrystals. So here we are using what? Some gold or tungsten particles. It will be coated with recombinant DNA. Okay, and it will be fired into the cell. Okay, so these are the what kind of gene transfer. Okay. Now we are coming to the next topic. That is the methods of selection of the recombinants. What is the method of selection of recombinants? Okay. So here there are two types of selection. One is known as the antibiotic resistance gene, and another one is known as blue-white selection. Okay, so we are talking about the selection of recombinants. Okay, so the basic principle regarding the selection of recombinants is that the insertional activation, the principle of insertional activation. That means it is a phenomenon of inactivation of a functional gene due to insertion of a foreign DNA. Just look at here. So this is our vector. That is, you know the name of the vector, that is PBR322. The vector. Okay. So these are the two antibiotic resistance gene. Okay. So if you are inserting a foreign gene in this area, if you are inserting a foreign gene in this area, that means what? BAMH1. It is an enzyme. The sequence of that enzyme. Okay. So if you are inserting a gene, a recombinant DNA, okay, a foreign DNA here, what will happen? It will fix it in this area, but the vector will lose what? The tetracycline resistance. Understand? So that is known as what? Insertional inactivation. Inactivation due to inserting the foreign DNA. That is known as what? Insertional inactivation. Okay. So here we are studying about the method of selection of the recombinants. Okay. How can we select the recombinants from the culture? Okay, by using our naked eyes, it is not possible. Okay, so that we are using what certain techniques. Okay, the first technique that is known as the antibiotic resistance. So when a foreign DNA is ligated or inserted into this sequence, BAMH1 sequence, what will happen? The tetracycline resistant gene, okay, it will lose its property. Okay. So we are using the vector what PBR322, and the resulting plasmids, it will lose what? The tetracycline resistance. So usually this vector is having tetracycline resistance. But if you are inserting something into this area, it will lose the property. Okay. So we have inserted a foreign DNA in this site, so that it has become what? A recombinant. Okay, a recombinant. The vector is recombinant. Okay. So what will happen is that now we are inserting the foreign DNA in this area. It will lose the tetracycline resistance. Okay. But it will have what? Resistance against ampicillin. Okay, so there are two genes. They are known as selectable markers: ampicillin resistance and the tetracycline resistance. Okay, so we are inserting foreign DNA here. It will lose the property. If we are inserting a foreign DNA here, ampicillin resistant gene will not work out. Okay, so here we are taking one example: what? Tetracycline resistance gene. Now it is inactive, but ampicillin resistance is here. Okay, now this cells, they are transferring into what? Ampicillin medium, a medium that contain what? Ampicillin. Understand? Then what will happen? All these can be what? Transformed. That means what? If we are incubating this structure, that is what? Ampicillin resistant and what? Tetracycline sensitive. It is sensitive to tetracycline because what? Some foreign DNA is already inserted into the tetracycline resistance gene, so it cannot work out. Okay, now we are allowing this to incubate in the ampicillin containing medium. So what will happen? They will survive because of what? There is ampicillin resistant gene is active. Okay, now we can say what? All these are transformed. Okay, these are it's very important. These are what? Transformed. Okay, so transformed means what? Sometimes it may be recombinant DNA, the desired recombinant DNA, or sometimes it may contain what? Self-ligated vector. That means that we have taken the BAMH1 enzyme, so it will make a cut. Okay, suppose none of the foreign DNA is inserted here. None of the foreign DNA is inserted here in this vector. What will happen? The vector itself ligate. At that time, the vector will not lose tetracycline resistance. That is what? Self-ligated vector. rDNA means what? The desired recombinant DNA. If the desired recombinant DNA is here, it will lose tetracycline resistance, but it is having what? Ampicillin resistance. Okay. If it is self-ligated vector, it will not lose tetracycline resistance as well as ampicillin resistance. Sometimes it may be incorrect recombinant plasmid. So if it is not desired DNA, it is not the desired DNA. So some of the undesired DNA has ligated in this site. Okay, it will become what? Recombinant, but it is not the desired gene. Okay, so that will also have what? Both of the resistance. Okay, so if you are dipping this in the ampicillin containing medium, all these are what? Transformed. They are transformed, but we do not know whether it is the desired DNA or whether it is self-ligated or whether it is incorrect plasmid. Okay, understand? Now, in the second step, we are transferring these bacterial cells with these vectors into what? Tetracycline resistance. Okay, the tetracycline medium. We are transferring into tetracycline medium. Then what will happen? Only the desired DNA, desired recombinant DNA will not survive. The desired recombinant DNA will not survive in the tetracycline medium. Why? Because already there, another gene is there now. The foreign gene is there, so that it will not survive. Then what about all others? That is self-ligated vector, incorrect recombinant plasmid. All these will survive. Okay, so in that way we can select the recombinant DNA. Understand? So here I told you that the selection of recombinants by using antibiotic resistance. We are taking two antibiotics, ampicillin, tetracycline. Okay, so in this tetracycline site, BAMH1 enzyme is there. It will cut there, and we are ligating a foreign DNA here, so that it will lose the tetracycline resistance. But if we insert this, or if we are dipping this into ampicillin containing medium, they will survive because they will have what? Ampicillin resistance. So they will survive. That means what? They are transformed. So they are transformed, but we do not know whether it is the desired recombinant DNA. So for that, we are transferring this into tetracycline medium. So only this desired recombinant DNA, they will not survive that in that medium. If it is not surviving, we can say that they are the desired recombinants. All others are what? Non-recombinants. Okay. So we can say that here one antibiotic resistance gene, that is ampicillin resistance. Okay. This gene, it helps to find out the recombinants. It will select transformants. It is used to select the transformants. The another antibiotic resistance gene, that is tetracycline, that will select the recombinants. Tetracycline select recombinants, ampicillin select transformants. Okay, all recombinants are what? They are transformants, but not all transformants are the real desired recombinants. Okay, the self-ligated vector, it is transformed. Self-ligated vector is transformed, but it is not what? It is not the real desired recombinant DNA. Okay, for selecting the recombinants, we need what? Tetracycline resistant gene. Okay, so that is the antibiotic resistance. Another selection is that the blue-white selection. Okay, so in this blue-white selection, so this technique, it will differentiate the recombinant plasmid from the non-recombinants by the ability of the non-recombinants to produce color, blue color, in the presence of chromogenic substrate. So here, the recombinant DNA. Okay, so we are taking that recombinant DNA, inserting that recombinant DNA into beta galactosidase enzyme site. That means in the LAC set gene. So the property of this LAC gene is that this LAC gene, it is responsible for the production of what? Beta galactosidase. Okay, but the usage of this beta galactosidase, it will convert the chromogenic substance into blue color. Chromogenic substance will be provided in the medium. Okay. Understand? So there will be what? A recombinant DNA, and that recombinant DNA, it will be inserted in the beta galactosidase enzyme site, that is LAC gene. If it is inserted, it will not work out. Okay, the LAC gene will transform this chromogenic substance into what? The blue color. Okay, so in that way also we can select the recombinants. Okay, so the recombinants will be developing what? No color. Non-recombinants will develop blue color. Okay, in that way we can select. So these are the two methods. Okay, so that's all about today, and you have to go through the textbook, and you can, if you have any doubt, you can ask me. Okay, now I'm going to put all unmute.